Good morning. Today is Monday, June 1st, 2020. My name is Ricardo Sandoval, and this video is for week 3 of Biology 225 Microbiology. This is the results video for Lab 4 Microscopy for Microbiology. And here's just a quick little slide I made here, just to kind of summarize what I'm going to be talking about. So in this lab, what I did was I went through the anatomy of my microscope. I viewed a letter E under all 4 powers, low, medium, high, and the oil immersion lens. I viewed prepared signs as well of bacteria and yeast under all 4 powers, and then determined which one was best for my viewing, and then took a picture of each. And then lastly, I created what are called 2 wet mount slides of my cheek cells and my dental tartar or plaque. So, I mean, as far as how this lab went, the most challenging portion that I found was trying to look at things through high power and the oil immersion lens. I mean, once I kind of figured out the coarse and the fine and the transverse and longitudinal knobs and kind of how everything works as far as the microscope was concerned, I did see the letter E quite easily when it came to the low and the medium powers. But when it came to high, I mean, I was very confused, quite frustrated for a bit, that all I could see were black lines. And I changed the coarse knob to make the microscope go up and down, and played with longitudinal and the transverse to make it work. I even switched the orientation of the slide, so I started out with the E facing me and the E facing the microscope, seeing if that would change anything, and it did nothing. And the same issue I had with the oil immersion lens. It just didn't show any real great amount of detail. Now, as I went through, or perhaps it did show detail, just, not what I was looking for as far as a solid E. But as I was kind of going through the lab and just doing a bit of more research, I realized that given, what the E looked like and kind of what I was looking for here, each lens did show what it was supposed to, but just because of the magnification being so high, 100 times on the oil immersion lens and 40 on the high power, it was not going to show me the actual E. So that was a bit of a learning experience. At one point, I thought that there was something wrong with my high power lens, but I later figured out that nothing was wrong indeed. So I also did view prepared slides that were already made, just so I could, again, practice with the magnification of the microscope. And those were of penicillium, yeast, spirillum, bacillus, coccyx, and also an amoeba. So, again, those, as I got more familiar with the microscope, those were relatively easy to see. Most of the slides I viewed best on either low or medium power, just because it made it, that was just easiest for me, again, just because of how high both the high power and the oil immersion lens were. So for the amoeba, I viewed it best under medium. The penicillium, I thought it quite well under high, which I was very surprised. Again, I thought at one point there was something wrong with my high power lens. The yeast and the spirillum I saw best under medium, and the bacillus and the coccus I saw best under low power. So, also kind of figuring out how to take a picture of the magnification with my phone was a bit of a challenge, but I figured out, because my hand sometimes tends to be shaking, I figured out kind of how to put one hand on the microscope, one hand on the phone, just to kind of stabilize. Now, the last part of this, I again created wet mount cells of my cheek and of my dental charter or plaque, which was quite fun, just to kind of see what my cells look like. So what I did to create the wet mount cells is, of course, I had, like my slides, two little square slide toppers, if you will, sterile cotton swabs, and a pipette with some distilled water. So the collection process, as you'll see in the previous video, in my experiment video, was relatively easy. I mean, it's been quite some time since I've created slides, biology many, many, many years ago. But it was very fun just kind of when I saw the slides to see what they look like. These weren't stained, so especially for both of them, honestly, the dental charter and the cheek cells, I did have to sort of kind of move the light blocks at the bottom of the microscope just so that the slides would be visible. But it was just really interesting to see no real problems with that. And then as far as the anatomy of the microscope, that went well. Thank you.